Thanks, Ken, and good morning, everyone. It's a great pleasure to be here, and I want to thank the organizers for this invitation and for the energy in the room and the progress that we're making. This is such an important area, and I hope that we can contribute a little this morning, Eric and myself, to the neurological aspects. I've extended the title of my talk from what's in the program to focus on Alzheimer's disease and the implications for treatment. We've developed ketone PET imaging, and I'm going to tell you a little bit about how we use that to try to establish the therapeutic potential of ketones in this important disease. I have some disclosures to mention. On the left side is my public and nonprofit funding, and you'll notice at the top we were talking about sources of funding. Some of it's gone from Canada to the US for some of the cancer work. Well, the American Alzheimer's Association has supported the trial that I'm going to talk to you about today. So it does go both ways. And there's some private sector support. And following Tim Noakes' experience that he so eloquently described yesterday, I want to mention in the bottom right there that I'm not a physician and I'm not providing any medical advice to anyone from this presentation. So we were talking, a couple of people have referred to Jeff Volek's Ford, and I just wanted to show a picture of it here. It's got a bigger reputation than it really is, and little do you know that it's actually a Toyota Prius, which is dressed up as a performance car. No, I'm making allusions to hybrid cars because I think it's a good analogy for the human brain, which is what I want you to think about in terms of its fuel requirements. There is an interchange normally between the ketones used by the brain and the glucose used by the brain, and this becomes faulty as we get older, and I want to tell you a little bit about how we explore that. And I want to start with the beginning. And the beginning is infant development. We've made allusions over the past couple of days to the importance of ketones and starvation, which is a secondary benefit of having ketones. But in fact, the infant brain cannot develop normally without ketones. So it's not an alternative fuel, it's an essential fuel to supply 20 to 25% of the infant's brain energy requirements day in, day out, physiologically. It's not a question of caloric restriction or intermittent feeding or whatever you like. It's a breastfed, healthy term infant. And part of that ketone supply to the brain is fueling the energy, but second point, is supplying carbon to make brain lipids, which is in the image there, which if any of you do brain imaging, you'll realize that's actually an adult brain rather, that's superimposed on a CT of an infant, or probably an ultrascan actually. And the white part of the image is the white matter, and the white matter is made primarily of lipids that are made from ketones, cholesterol, long-chain fatty acids, the myelin, the sheaths around the axons, and the nerves. So it's an important anabolic substrate for the developing brain, and it's an important catabolic or fuel substrate as well. The main source of the carbon to supply the ketones that are constantly coming into the infant during lactation is the medium-chain triglycerides that are in milk. Human milk, all mammalian milk that we know of contain medium-chain fatty acids, and in principle mean that the infants of most or probably all mammals are in fact in mild ketosis during the lactation period. The advantage in humans is that some of those medium-chain triglycerides end up in storage fat, in the body fat of the infant. We are unique as a mammal in having body fat at birth, and so this in a way extends the ketogenic state beyond weaning, because those MCT can then be used from body fat. And I've discussed this in the implications of this for human evolution, both in the book that Ken referred to and in this article in the Journal of Human Evolution a couple of years ago. So I think the important point I want to make here is that what we're trying to do is recapitulate in the aging brain what the newborn brain is used to and thrives on day in, day out. The second, I think, important point that I want to make is that the fuels that are used by the brain, there's two different strategies. I notice on my screen that it's partly cut off, but I'm glad to see it's not cut off at the top for you. So the two strategies are that the glucose gets into the brain because it's pulled into the brain. This little stick person is pulling a glucose molecule into the brain, which is on the right. And that process occurs through the glucose transporter, and it's driven by a fall in glucose in the brain cell that's doing some job, communicating with its neighbor. So this is a process that's driven by brain activity. That's strategy number one. It's still supplying most of the fuel that the brain is going to use. But the alternative strategy is driven by ketones, and it's a push strategy. So when the ketones go up in the blood on the left, they are pushed into the brain. And this is the way the brain works all the time. If glucose goes down because you've been fasting for 24 hours, for argument's sake, insulin is down and ketones are being produced, and they will go into the brain in relation to the concentration as shown on this slide. So this is a compilation of a number of studies which I've compiled in the publication that's noted. And you'll see on the x-axis the plasma ketones are on a log scale, and the fasting indeed goes from the postprandial to 24 hours fasting, 40-day fasting, which was referred to George Cahill's classic work, but there is also a paper published in the Journal of Clinical Investigation on a 60-day fast in obese individuals as well, where you get over 10 millimolar beta-hydroxybutyrate in the blood, and you're supplying, as you can see on the y-axis on the right-hand side, about 85 to 90% of the brain's energy requirements. Obviously, it's an extreme situation, a non-physiological situation, probably ethically impossible to do today. All I'm using it for is to demonstrate the principle that as ketones become available in the blood under these circumstances, they will be used by the brain. So there's a linear relationship over a 600-fold range at least, which is an extraordinarily efficient way of providing a backup fuel for the brain. The third point I want to make is that glucose is the main fuel of the brain under most circumstances, except under extreme fasting, but it's not the preferred fuel. So there's a difference between the main fuel and the preferred fuel. And this is an example of a study that we did with a PET scan. I'll show you how we did the PET in a minute, but I wanted to set the stage for the Alzheimer's studies with these couple of preliminary points. So on the x-axis, you can see a change in brain ketone uptake caused by the ketogenic diet. So these are delta values. They are increasing amounts of brain ketone uptake compared to the pre-ketogenic diet stage. And on the y-axis is the glucose uptake, which goes down in proportion to the increase in ketone supply. So glucose uptake is going down when there's sufficient ketones around to go into the brain. So this is an inverse relationship, and it's why I refer to ketones as being the preferred fuel. We're not the first people to have shown this, and there's a very useful glucose-sparing effect that, in fact, occurs on the ketogenic diet. So the context for this in relation to aging and Alzheimer's disease is shown by these two little arrows. And if you look at the two scans, you can see that the one where the arrows are has basically missing the orange to red color over the ears, the parietal lobes. This is a classic image of what one sees in Alzheimer's disease, and it's been known since the dawn of PET scans in the late 70s. The interpretation has almost always been, well, the brain cells are dying in Alzheimer's disease, so they don't need glucose, so the glucose uptake goes down. And that's a logical interpretation, except for 2 points that I want to make. The first point, and it's in this concept that we've developed over the past couple of years, along with some other people around the world, is that in fact the glucose problem is specific to glucose and it precedes the cognitive deficit in Alzheimer's disease. So how do we know that? What we're talking about is still the definition of the glucose deficit is defined by the PET scans where the color, the reddish color, is declining, and that's showing lower glucose uptake in the brain. In the yellow box, there are 5 categories of people with presymptomatic brain glucose hypometabolism, and they're all categories of individuals at risk of Alzheimer's disease. Who are they? Older people, people with insulin resistance independent of age, people with a family history of Alzheimer's disease, people with, that are ApoE4 carriers, and the presenilin mutation. So those are very different groups of people. They've all been identified as having a risk of Alzheimer's disease, an elevated risk of Alzheimer's disease, and they all have presymptomatic brain glucose hypometabolism. So this cannot just be a consequence of the disease. They don't yet have the disease. They're all cognitively normal. They're at risk, but they are still cognitively normal when the PET scans have been done. I haven't given you the citations. I'll be happy to, if you look up our work, all this work has been referenced and has been done by other groups. So the way we look at this now is that this latent glucose hypometabolism is in fact helping precipitate the neuropathology associated with Alzheimer's disease, deterioration of synapses, and deterioration in brain function, which is associated with cognitive decline and which is going to push down the glucose metabolism further and is going to create a vicious cycle of brain energy exhaustion and progression of the disease. So we could argue about whether the neuropathology comes before the latent glucose hypometabolism, but we have a way of assessing this and saying, if there are two fuels that the brain can use, what about brain ketone uptake in people at risk with Alzheimer's disease or at risk of Alzheimer's disease? If it's the same pattern as it is with glucose, then we know the neuron's completely screwed up, or the astrocyte, because it's a different transporter mechanism, it's a different access point to the Krebs cycle, it's a different pathway until you get to the common pathway. So let's explore brain ketone uptake in these conditions. So how do you do that? You do what we've called dual tracer quantitative PET imaging, and the protocol is shown here. We do the PET image with a ketone tracer, which is acetoacetate, carbon 11 labeled, short half-life, so you have time to collect the image before the radioactivity is decayed away completely. And then you have a washout period, and then you do the classical PET FDG scan on the same individual on the same afternoon. They're in the same physiological state, so the images are quite comparable. You can do this qualitatively with what I think Parker referred to SUVs, standardized uptake values. It's a relative measure, but it's not good enough if you're trying to figure out what the problem with glucose is, the extent of the problem, what your therapeutic target with ketones is. You need to quantify these scans. And in order to do that, you need blood samples, and you also need an MR image to localize the PET image and get the exact information about what regions are affected and what the extent of the glucose problem is. So this is what the scan is. You can see the references. We've published a few papers now. We've studied over 300 people using this methodology. And the units are classically known as CMR units, cerebral metabolic rate. This is a quantitative value with micromoles per 100 grams per minute. And there are basically two units to this calculation. One is an uptake rate constant, K, which is taken from the PET scan, and the other is the plasma ketone value. And as many of you know now, plasma ketones can vary a lot if you're on a ketogenic diet or if you're on a ketogenic supplement. So the results become fairly noisy. But the uptake rate constant is actually a fairly tight value depending on the conditions. So if we look at the people that are of most interest during brain aging and Alzheimer's disease, the CTL are cognitively normal, healthy, older people, age matched to the MCI, which is mild cognitive impairment, and AD Alzheimer's, and we've got about 20 people per group in this project. And these are the K values. This is the capacity of the brain to take up the ketones. It's kind of like how many doors are open. If someone calls fire here, if there's only 2 doors that open, we're going to back up before we can get through everyone through the doorways. If 4 doors open or 6 doors open, that's a larger K value in the analogy of fire and trying to escape. And as you can see here with glucose, the capacity of the brain to use glucose is higher in the areas where the red and orange colors are shown on the brain. And as you go towards Alzheimer's, you can see there's a virtual disappearance of the red, orange, and the green is more predominant. So the capacity of the brain to use glucose decreases in Alzheimer's disease. And this has been established for many, many years. If we look at the ketone uptake in exactly the same individuals, you can see that the green color, in fact, the capacity is lower than it is for glucose under the normal circumstances, but it, if anything, actually increases as you get towards Alzheimer's disease. There is no loss of the capacity to transport ketones into the brain. It's the same cells, so they can't be dead because this is an active transport-mediated process. So this is encouraging us to believe that we could use the normal brain ketone uptake capacity to, in fact, bypass the glucose deficit and potentially have an impact on cognitive function. So the issue then is, well, that's basal uptake rates. What about uptake rates when you provide a ketogenic substrate? Simple way of doing this is with an MCT, a medium-chain triglyceride. We arbitrarily hit on a dose of 30 grams a day. We did this over 1 month. And this rather busy-looking slide is basically showing you that the plasma values are on the x-axis, and the star and triangle are before they start the treatment. So the values are lower, around 0.35, 0.4 total ketones. When they go on either of these two MCT supplements, we basically double the plasma levels, and the brain values are shown on the y-axis, and you can see that we've doubled the brain uptake on the y-axis. The key values are in the background. The dotted line is what the reference value, the slope of the line that we would ideally like to see, that has been shown in our studies with young healthy adults. And basically, they're identical, statistically identical slopes for the Alzheimer patients. If we were to extend these plasma values to much, much higher values, we might see that the slope of this line connecting the blue triangles, the green triangles, might drift off, but it's very close to what it should be. So this suggests that the brain can use additional ketones in Alzheimer's disease when you provide them. So the important thing then is, well, what happens to memory and cognition? And we've done a study in mild cognitive impairment. We call it the BENEFIC trial, which is for brain energy, functional imaging, and cognition. And the cognitive outcomes are summarized on this slide. This study was designed and powered for the metabolic outcomes. Is the capacity to use ketones normal in mild cognitive impairment? And the answer is yes. It's the same results as I just showed you for Alzheimer's disease. But we still have 19 or 20 people per group, and we had a placebo control on this. So we have cognitive results, and you can see that there are several domains of cognitive function that improve, particularly episodic memory, but also processing speed and language that do not improve on the placebo. So this is encouraging, but it's not definitive, because a clinical effect with a cognitive outcome would require probably closer to 100 people per group. Importantly, though, with the brain ketone PET, we can show that there's a statistically significant positive relationship between the plasma levels or the brain ketone uptake and the outcomes on cognitive function. And this is one example with the quite commonly used trail-making test, on which there was a positive effect in mild cognitive impairment. And you can see that there's a linear positive relationship in relation to the brain ketone uptake. But it's also clear that the placebo group and the active group are not well separated here at all. So the ketone effect of the MCT does not really pull them, separate them from the placebo group at 30 grams a day, which I don't have on the slide, I see, but that's the dose we use for 6 months. So that's something that we now realize is helping us set a therapeutic target. We arbitrarily started with 30 grams a day, which was close to the upper tolerable limit, we thought, with the formulation that we had. And we were trying to demonstrate a metabolic effect, and we've seen that metabolic effect. But now, if we expect to see a cognitive effect, we're going to have to increase the brain ketone supply a little more. Still, the trend is there, and as the yellow box says, several other of these correlations were also positive, but I don't have time to show them to you. So again, proof of principle suggests that you can achieve a metabolic rescue of the brain, and that the rescue is going to have a functional effect that is going to correlate with the level of ketones achieved both in the blood and in the brain. So we look at this now in terms of the green band in the middle here. What is the energy rescue, or the gap in energy supply to the brain that we're trying to fill? So let me take you through this. The bar on the left here is the young, healthy controls. And their energy supply is there in the normal group, so they're at 100%. And their brain energy is coming, as you can see in the table at the bottom, about 95% from glucose and about 5% from ketones, using the dual tracer ketone PET imaging. So that's the young, healthy controls. Older, healthy controls, we got about 30 people in that group, and you can see that the total height of the bar is lower. The ketone contribution is a bit lower. It's not that the K value for the ability to take up the ketones goes down, but in fact, plasma ketones drift downwards as you get older. So their contribution to brain energy metabolism goes down. But it's glucose that is going down in principle here, mostly from 95% contribution to about 89%. But they're all cognitively normal still. Then you go to MCI, which is the middle bar, and the value has gone down a little further. So there's still a significant drop in glucose, although the ketone uptake is still the same as it was in the healthy older people. Then the second bar from the right is the trial I just mentioned, the BENEFIC trial, in which the MCI patients were on 30 grams a day of the MCT. And now we've helped restore them closer to the healthy elderly with the ketones, which are now contributing twice as much as they were in the healthy elderly, but the glucose contribution has not been changed by the MCT supplement. So this is suggesting to us that if we want to get a more complete reversal of the energy deficit, we're going to need to get into 45 grams a day, roughly speaking. And that was going to put us somewhere between the healthy elderly and the healthy young, which seems like a reasonable place to be. We might be able to get right up to this 100% value perhaps with a ketone ester or some other supplement. We're still exploring that. So this is telling us what our therapeutic target is. It's giving us a dose that we should be looking for in order to accomplish this particular goal. This is the same slide, but it's a little busier now because I've added the Alzheimer's group here. So from the MCI, we get a further decline to a value of a deficit of about 19% in early Alzheimer's disease. So a person with early diagnosis of Alzheimer's disease, day in, day out, is facing a 20% energy deficit in their brain, constantly. MCT is one way to help correct that, as we show you here, but you can see that the group next to the Alzheimer group here is an exercise group. And in fact, exercise helps get glucose more into the brain. It also helps get ketones into the brain without a ketogenic supplement at the same time. It's helping to increase the K value, the number of doors into the brain. So exercise is potentially quite complementary with this, and we're excited about the potential of that. The problem is that it took 7 years to get 5 patients through this protocol. So it's not easy to get Alzheimer patients to participate in these sorts of projects, but the proof of a beneficial effect is there. A second Alzheimer group, which are basically identical to this one on 30 grams a day, is also improving the brain energy status, but it's not changing glucose. So medium-chain triglycerides are not affecting glucose. Exercise is affecting both, and I think together we could probably catapult them just over this magical line here. So it's kind of exciting to know that we can do some things about the brain energy status in Alzheimer's disease, and we need to power studies now for the cognitive outcomes. So coming back to that busy slide with the direct line relationship between in normal young adults who have been starved or not, that's the black line that's shown here, and what I'm showing next to it is the uptake, not of beta-hydroxybutyrate, which is the black line, but the dotted line, which is acetoacetate. And I've put the Alzheimer patients on there, I've put our healthy older controls on there, and I've put the ketogenic diet on there, and they're all in the same linear relationship. So it's very achievable. People who are on a ketogenic diet are probably at least at 1 millimolar ketones in the blood, and this should be really helping to stave off, if you're in a preventive mode, the glucose deficit in the brain. But this is what we imagine is probably the target that we're going to need on a day-long basis, 24 hours. It's very hard to achieve with a medium-chain triglyceride supplement, but much easier to achieve with a ketogenic diet, although compliance is tougher in older people. So just to show you what a ketogenic diet does to the brain ketone uptake, these are the cerebral metabolic rates for acetoacetate. And the blue color is in people pre-ketogenic diet. Very low ketone uptake, around 5%, and that's what you would see. The blue represents quite a low uptake, and then everything basically lights up when you're on a ketogenic diet. So those of you that are on a ketogenic diet, you can see how your brain loves the ketones. And in fact, the ketogenic diet is actually increasing the total supply to energy of the brain. This is in a normal adult where there's 5% ketones, 95% glucose, around 100%. There are two different control groups here for reasons that I don't need to explain. They're both about 100, but in fact, you can get to 113% or so on a ketogenic diet. There's more energy getting into your brain on a ketogenic diet than there is on a typical Western diet. So there's not just a therapeutic effect, but potentially a cognitive benefit in those that are not facing aging-associated cognitive decline. So to summarize, I just want to come back to this image and say that those arrows are definitely a consequence of the disease, but they're also contributing to the disease. They are present, the arrows are present before the disease starts, and it's a glucose-specific problem. That brain energy rescue by ketones is definitely feasible in mild cognitive impairment. It's feasible in Alzheimer's disease. What we're trying to do is basically let the brain have the luxury of thriving in a fuel environment that you were born into. And the fourth point is that the cognitive benefits still need to be better defined, as well as the mechanism of action. We don't know if it's, as alluded to earlier today, we don't know if it's just a fuel effect or whether there's some signaling effects, whether there's some anti-inflammatory effects, whether even affecting the pathology in the brain in this situation. So I want to finish by thanking a terrific group of collaborators and members of my lab, and thank you for your attention.